Hi, welcome to our YouTube channel. Thanks for subscribing to our channel. Really appreciate you. I will quickly want to look at the rundown of the 2024 Waec Agric Science Practical. The Waec 2024 Agric Science Practical Specimen. So those are the things that we quickly want to look at. And so here we have listed out all of the specimens as they have been highlighted or selected by Waec. So for specimen A, we have the ranging pole. Specimen B, we have the arrow. Specimen C is the measuring tape. Specimen D is wooden peg. Specimen E is the dry sand in capillary tube, while we have specimen F to be dry clay in capillary tube. We have the specimen G to be loamy soil in capillary tube, and then we have our specimen H to be the maize weevil. Specimen I is the bean beetle. The specimen J is grasshopper, while we have specimen K to be cotton stainer. Specimen L is tilapia. Specimen M is hay, and the last one, which is specimen N, is the fish meal. So we'll quickly look at some characteristics of all of these specimens as possible questions that you might be coming across in your examination, and we'll show us some possible pictures of what we are talking about so you get familiarized with all of them. Now we are moving to the first one, which is our specimen A. For our specimen A, we have it to be the ranging pole. Specimen A is the ranging pole. Now, the ranging pole is actually a survey instrument that usually has a thickness or a diameter of 3 to 4 centimeters, and then it has a length of 2 to 3 meters. The length of the ranging pole is usually about 2 to 3 meters. They are usually characterized by several colors. Basically, you can have them to be red and white, or you can also have them to be black and white. Those are the colors combinations that you can have for the ranging pole. It is basically used for marking the positions or the boundaries of stations. When you are on a field carrying out survey, you actually use the ranging pole to map out locations, boundaries, positions on that field. Also, it can be used for sighting in the station. It can be used for sighting on the stations, as well as it being used for ranging straight lines. Those are the characteristics that we need to understand about the ranging pole. If I quickly decide to show us some pictures, we'll be having something that looks like this. This is an example of a ranging pole. We can see this now. It is characterized with black and white color alternating at specific measurements. You can see them here. This is black for 0.2 meters, after which it has another 0.2 meter of white, then black again of 0.2 meters, and it keeps alternating like that. We can have another one like this. One is black and white. We can have another one that is red and white. You can see this one here. So that is basically what we are talking about. You can see the total length that we are having here. Like I said earlier on, that the length of the ranging pole could be 2 to 3 meters, while the diameter, that is from this side here to this side here, will be about 4 centimeters. So that is basically what the ranging pole is. The next specimen, which happens to be the arrow. The arrow is also another type of surveying instrument, and it can also be called the marking pin. That is another name that is oftentimes used to represent or to identify what we refer to as the arrow. It is also called the marking pins. Now, it is basically used in large flat surfaces or land to mark the ends of the chains during chaining process. Yes, when we carry out chaining process, we basically use this particular marking pins to pin the areas or the locations where the chain is ending before we run the chain through another session. As well, you pin another marking pin at the end of the chain again, and you consecutively do that until you get to the end of the field where you are carrying out your survey. So you now count the total area or the total number of marking pins that you have pegged into the ground. Basically, that is what is done. So the description: the pins are arrows, or the pins or the arrows, they have about 4 millimeter diameter. They are 4 millimeter in diameter and can have a length. They can have a length of about 500 millimeter. Their length could be about 500 millimeter. It also has a loop at its end, which has a 50-meter diameter. The loop at the end of that marking pin oftentimes or usually is 50 millimeter in diameter. Now this loop has its function. The loop has its function. It is basically there crafted into the arrows to make it handy, to help make it durable, to carry it about, and it is also used to pick them up quickly as well as stringing them together using wire or cords. So if you decide to look at what this specimen is, let's see what it looks like here. So this is an example of a marking pin or an arrow. You can see it. You can see the loop at the top of it here. This is where we have the loop. So with this we can actually handle what the marking pins or the arrow are. So we pin this into the ground, having this at the top. So that is an example of a marking pin or an arrow. This is another example. You can see the loop clearly shown at the top of the arrow. You can see them here. This is a 10 set marking pin or arrow. So that is that about the specimen B. So we move to specimen C. Specimen C is the measuring tape. The measuring tape is the specimen C, and it is a handy tool typically used to measure distance, length, or the size. It is much larger, longer than it can be seen as a meter rule. It is oftentimes seen as a flexible ruler. It consists of a case. It could have a thumb block, the blade, or the tape hook, as well as sometimes it can have belt clip. A tape usually has an imperial reading, and sometimes it could have metric reading, or in some cases it will have the two together. It will have the imperial reading as well as the metric reading. So when we look at that, we will be having some types of tapes looking like the ruler. So when we want to look at such, this is what we will be seeing. We will be seeing something like this. This is an example of a measuring tape. Now this part of the measuring tape here is graded in inches. You can see this part here graded in inches, while this other part there, this reverse side here, is graded in centimeters. So we have so many types of tapes. This is one type of tape. Here we have another type of tape. You can see this is another tape. This one is much longer than that one that we just talked about, or that we just shown, that has just been shown. So those are what tapes looks like. This part here is measuring in centimeter. This part here is measuring in centimeter, as opposing this other side that is measuring in inches. So basically that is what specimen C is talking about. And if you look at specimen D is a wooden peg. A wooden peg that is also called a survey stake. It is also called survey stake because it is actually used to map out locations or boundaries on the field. It is usually made of hardwood. Since it is called the wooden peg, it is made of hardwood, and it indicates points and positions on the site. Sometimes you can have some of these pegs made of some other materials, like it could be made of metals, it could also be made of plastics. But the one we are talking about here for this particular experiment is the wooden peg made of hard wood. They are usually of different sizes and have colored tops. They are of different sizes and they often have colored tops. So we could have them looking like this. This is an example of what we refer to as a wooden peg. You can see the top of it here having a red color, so it makes it visible from afar when you have dug it, or when you have struck it into the ground. So this red patch here makes it visible from afar. That is what the marking pin is made of, and that is what it is used for. This is another example of a marking pin that has been inserted into the ground. You can see it clearly here. So it has been inserted into the ground. You can see it here, inserted into the ground, and it has a reading on it with the color that is on it. So that is basically what the marking pin, the wooden peg rather, is about. So we move to the next specimen now, which is specimen EFG. Specimen EFG, they are a set of specimens. We have them to be a set. Soil samples. Specimen E is dry sand in capillary tube. Specimen E is dry sand in capillary tube. We have specimen F to be dry clay in capillary tube, and specimen G is loam clay in capillary tube. We are actually using this particular experiment or this particular set of specimens to carry out a specific experiment on soil, trying to know their water retaining capability or capacity. That is basically what we want to look at. So we can see here that we have the capillary tubes. Let me just quickly explain how it is constructed. We have capillary tubes. Let's assume this is we have capillary tubes A, B, C. Supposedly we have E, F, G. Particularly for this experiment, we have the capillary tube E, F, and G. We have the E to be sand, and then we have clay and loam. That is the setting, like what we have here: sand, clay, and loam. So we eventually found out that in the setup, like it was prepared here, at the tip of the capillary tube, it was plugged with cotton. You can see each of the capillary tube here has cotton at the end of it, the cotton wool at the end of it, after which the sand, the soil sample was poured into the capillary tube, and it was allowed to go down into the base of the capillary tube. So in this first tube here, according to this experimental setup, we have loam soil, here we have sandy soil, and here we have clay soil. Now the depth, they decided to insert all of these capillary tubes into a basin or a trough of water. Now in the capillary tube will start permitting the soil sample in each of the capillary tubes will start absorbing water, and the water starts to move up in the capillary tube. Basically, that is what the experiment is talking about. So capillarity is the ability of various soils and rocks to allow water to move up through the capillary tube. Basically, that is what the experiment is looking at. It is dependent on the soil types. Yes, the ability of the water to move upwards in the capillary tube is dependent on the soil type's porosity as well as its permeability. Now there are some forces that are responsible for this taking place. The forces involved in the capillary, they include, that is gravity. It pulls water molecule through the soil types, and then capillarity is the primary force that enables the soil to retain water as well as to regulate its movement. So basically that is what capillarity in these soil sample cases is talking about. And then we move to the next experiment. Okay, let me quickly show us. So in this experiment now, we discovered that clay soil absorbed the most amount of water, and that is why we see the level here. You can see the level of water here. In clay soil, it absorbed the most amount of water, followed by loamy soil, and sandy soil happened to have or have absorbed the least amount of water from the trough. That is what we have in the two cases. So we can see what is happening here as well. The clay soil has absorbed most amount of the water, followed by loam, which has absorbed considerable amounts, and then the sandy soil has the least amount of water absorbed. So basically that is it. And then we move to the next one, which is specimen H. Specimen H actually is the maize weevil. Specimen H is the maize weevil, and its botanical name is the Cytophilus zimes. Cytophilus zimes, like we have seen it here. Now it is a species of the beetle. It's a species of the beetle, and it actually is destructive. It attacks and damages or destroys standing crops as well as stored cereal products like rice, sorghum, and the rest of this. It doesn't affect only the maize, it also affects some other materials or crops like rice, like sorghum. It affects them while they are on the field and when they are being stored in their silos or in their containers. It also infests other types of stored processed cereals like pasta, like cassava. And here we have the classification of this particular crop. We have it, since it is an animal, it belongs to the kingdom Animalia. Then the phylum is the Arthropoda, the class is the Insecta, the order is the Coleoptera. We have the family to be the Coculeonidae, and then the genus is the Cytophilus, while the species is the Zimes. So that is the classification or the taxonomy of this particular organism called the maize weevil. Now it has complete metamorphosis, it undergoes complete metamorphosis. That is, It moves from the egg to the larva to the pupa and to the adult, and it takes about 36 days for the egg to mature fully into an adult. And then the adult maize weevil can live for about 5 to 8 months. That is their lifespan. They can live for about 5 to 8 months. It is very destructive, hence it reduces agricultural yield and benefit. They are very very destructive, hence they reduce agricultural yield and benefit. And they have four pairs of working legs. So let me quickly show us example. So this is the example of the maize weevil, the Cytophilus zimes, the Cytophilus zimes. So this is the example of what we have there. We can see now that it has four pairs of working legs. This is one. We have 1, 2, 3, and 4. So this is another diagram that can help us see it clearly or more clearly. We can see it here. We can see all the working legs now. 1, 2, 3, 4. Those are the pairs of working legs that we have for this particular specimen. So it is advisable that we get ourselves familiar with these diagrams to be able to answer questions. Peradventure we come across any of them, we can see them. So that is that about this particular specimen. And then we move to the next one, which is specimen I. Specimen I is another insect, that is the bean beetle. The bean beetle, and its own botanical name, or it's a group name, we have it to be the Bruchine species. The Bruchine species has so many species. Example of the species that we have there, we have the Acanthosidis obtecus. We also have the Calosobruchus maculatus. We also have the Calosobruchus chinensis. These are different species of this particular bean beetle, and we have here listed as out here the classification of this organism. So we have the kingdom Animalia, we have the Arthropoda, we have the class to be the Insecta, the order is also Coleoptera, and then the family here we have it to be the Chrysomelidae, and then the genus we have it to be Acanthocelides, as well as the species being Obtectus. So this is another species, like I have written it out here. So this particular organism has 3 pairs of working legs, like we will see very shortly. It has 3 pairs of working legs. They primarily feed on seeds of common bean and some other crops like the pea, as well as legumes. They are pests because they destroy plants both on the field and in storage. Only the larva feeds on seeds. Only the larva of this particular bean beetle feeds on seed. The adults feed on pollens. A single adult female of this beetle can actually lay up to 300 eggs during its lifetime, and it also carries out, or it also undergoes complete metamorphosis. Also, this one also undergoes complete metamorphosis. So this is an example of a beetle, a bean beetle. This is an example of a bean beetle, and we can see its metamorphosis here now. It lays eggs, and from the eggs it matures into the larva stage. This is the young larva, the full grown larva, before it goes into the pupa stage and then matures fully into the adult stage. So that is what the bean beetle looks like. Labeled diagram. So this is what it looks like. We can see the diagram well labeled. So we need to familiarize ourselves with this diagram to make it possible for us to actually answer questions when we come across them. So that is what this particular specimen looks like. And then we move to the next specimen, which is specimen J. Specimen J also is another insect, that is the grasshopper. It is its own group name is the Califera species. It is the Califera species. We have other species like the Melanopus differentialis. We have the Melanopus differentialis. We also have the Homocestus viridulus. The Homocestus viridulus. Those are also those are other species of this particular organism or specimen called grasshopper. Now, it has its own classification as well. It is from kingdom Animalia. The phylum is Arthropoda. Class is Insecta. The order is the Orthoptera. the other is Orthoptera. We have a family to be Acrididae, and then the genus happens to be Homocestus, and then the species for this particular one we are looking at is the Viridulus. That is the species of this particular organism, and that is why we have the botanical name of this particular one to be the Homocestus viridulus. So what do we need to know about this organism? They are chewing herbivorous insects. They are insects that are herbivorous. They actually chew or feed on plant parts. They are grouped and dwellers. They are ground dwellers. They possess powerful hind legs or legs, which makes it possible for them to actually evade threats from their predators. And they also have forelimbs that is used. Those forelimbs are usually shorter and they are used to grasp food. They sometimes are seen as pests of cereals. They are sometimes seen as pests of cereals, of pastures, as well as the vegetables. They protect themselves from predators by camouflage because they actually conceal themselves in their environment, thereby hiding or protecting themselves from predator, preventing themselves from being seen by the predators. This type of organism has incomplete, or it undergoes incomplete metamorphosis, where the egg develops into nymph and then the nymph develops into adult, and that is what is seen in these grasshoppers. Now when they fly in bunch, when they swarm together, grasshoppers are seen as locusts. When they swarm in multitude, they are seen as locusts. That is what the grasshopper is. So this is an example of what the grasshopper looks like. We have this here as a grasshopper, and this is another diagram of the grasshopper, a labeled diagram of the grasshopper. So we can see it has the compound eye, it has the simple eye, it has the antenna, it has the thorax, the abdomen, it has very powerful limbs. You can see the limbs here. The limbs are very, very powerful. With it they are able to jump and to move rapidly, and we can see other body parts here along with the wings. So that is basically what the grasshopper looks like. So we move to the next specimen now. The next specimen, which happens to be specimen K, that is the cotton stainer. The cotton stainer, and its botanical name is the Disdecus singulatus. The Disdecus singulatus is the botanical name for this particular organism. It is also having multiple species. So we have another one which is the Disdecus citrocellus. We have another one which is the Disdecus citrurellus. So we need to understand this. Now, looking at the classification, we have the kingdom Animalia from the phylum, which is Arthropoda. Sorry, this is supposed to be Arthropoda here. And then Arthropoda, and then we have the class to be Insecta. The other is the Hemiptera. We have the family to be the Pyrrhocoridae. We have it to be Pyrrhocoridae, and then the genus is the Disdecus, as well as the species being Singulatus. So we have so many of them. Like we have another species which is the Disdecus citrurellus. So this particular cotton stainer derives its name from its habitual effects on cotton. Its name is derived from its habit on cotton because it actually punctures and sucks the young balls of the cotton, thereby preventing them from reaching maturity. When it sucks the balls of the cotton, it prevents the cotton from actually producing enough oil, as well as it having the night color that it should have. It also destroys the color of the cotton, colorizing the cotton, so it stains the cotton. Now this particular pest also affects some other type, or this specimen rather, it affects some other crops. It also affects oranges, and when it inserts its proboscis into the orange, it inserts some chemicals into the orange, thereby causing the chemical to drop in few hours from its tree, and in a matter of 1 to 2 days that orange fruit rots away. So it can be controlled. This particular cotton stainer can be controlled. And how do we control the cotton stainer? It can actually be controlled using soapy water. You can actually spray it using soapy water, or you use particular or precise amount of insecticide. Those are the things that can be done to control cotton stain. This is what the cotton stainer looks like. This is an example of the cotton stainer. It is actually there to prevent cottons from actually attaining maturity or reaching full maturity. It stains the cotton as well as destroys the ball of the cotton. So this is a cotton stainer, and this is a labeled diagram of the cotton stainer. You can see the parts of the cotton stainer. So that is basically what the cotton stainer is about. And then we move to the next specimen, which happens to be specimen L, which is the tilapia spermanis. That is the botanical name, the zoological name rather, tilapia spermani. It has its own classification, like we can see here. Kingdom Animalia Chordata, Kingdom Animalia, then the phylum is Chordata, the class is Actinopterygium, and then we have the order to be the Cycliformes, while the family is the Cichlidae, and then the genus is Tilapia, while the species is the Spermani. So this particular organism has its own adaptive features, structural adaptive features, that makes it possible for it to survive in its environment. Like it possesses gill system for respiratory functions, for gaseous exchange. It also has the streamlined bodies that makes it possible for it to swim and to pass or to move around in the waters. It has scales that prevents water loss to the surrounding, maintains water balance in the body of the organism, as well as fins, which makes it possible for it to also swim in the waters. This particular organism feeds on algae and zooplankton, particularly the smaller tilapia fishes. They feed on algae and zooplanktons. Now, there are organisms that are actually found in fresh waters. You will rarely see them in salt water or brackish waters. They are usually found in fresh waters like ponds, like rivers, and the rest of those. And they can live up to 10 years and can reach a weight of about 10 pounds. They can actually develop to weigh up to about 10 pounds. And one of the healthiest sources of protein is this tilapia fish. It has numerous amounts of protein in it. The adults can be omnivores. The adults of these fishes can be omnivorous at times, where they feed on smaller fishes as well as planktons in the waters. And sometimes they can become cannibalistic, particularly when there are no things or food samples for them to rely on in that environment. So this is what the tilapia fish looks like. We can see it having the streamlined body. So let me show us. This is another example of the tilapia. This is the label diagram of the tilapia fish. You can see the fins here. The fins are shown here. We have the dorsal fin here, and then we have the caudal fin here. We have the anal fin here. It also has the pectoral fin as well as the pelvic fin. It has the eye, the mouth, the baculum around here. So these are parts or body parts of the fish. It has scales all over it here. So you can see that is what the tilapia fish looks like. Now, in cases where you want to differentiate between the male and the female, the sexes of this fish, this is how the male genital looks like, and that of the female looks like this. So those are the things that we need to know about the tilapia fish, which is specimen L. And so the next specimen we have there is specimen M, which is hay. Hay is the next specimen. Hay is actually made from the combination of grasses, legumes, and other herbaceous plants. They are cut and dried before being used as fodder or food for livestock and cattle and sheep, etc. Some other smaller animals like rabbits also feed on hay. Now, these hays are produced basically because some livestock do not get the ability, or they do not get the opportunity to graze on open fields. So when they are not opportune to graze, they will require to be fed by some amount of hay, and that is where we have the major processes here. So the major processes that we have the cutting, we have the drying or the curing. After cutting is done, they will dry the hays and then they rake them, after which they actually process them before they are being stored in barns. It is best served, this particular hay, they are best served to have their good quality when the leaves are still green, and it should actually not be too coarse. It should not be too coarse. you should have enough plant leaves, heads and stems. So when we look at them, this is what a typical hay should look like. So this is what the hay looks like. We have the hay here. This is a hay that is freshly cut and is just being processed. So they are not yet dry to a level that could have resulted them to becoming like this. This is another type of hay. So basically that is what it is. They are fodder or food for the uh, for the animals, specifically the herbivorous animals. And then we have our next specimen, which happens to be the last specimen, to be specimen N, the fish meal. Specimen N is the fish meal. It could also be called fish protein concentrate. They are usually made from large catch of fishes. These fish byproducts are used to feed farm animals. Example, they use them to feed pigs, poultry, also some farmed fishes. They contain small oceanic fishes such as the herrings, the sardines, salamanders and the rest of those. The oil and the water are pressed out, after which the remaining solid is cooked and pulverized into a meal. This fish meal is also important to humans because humans also feed on them because of their nutritive value. They have very high nutritional content in them. As such, humans also feed on them. They are rich in high quality proteins, essential minerals, acids, amino acids, vitamins and minerals. So this fish meal, they are very good for protein supplements. They are good for protein supplements. So this is example of what we are talking about. So this is an example of what we are talking about. This is the fish supplement that we have, the fish meal. So this is the fish meal. We also have this one also to be fish meal. This is another type of fish meal. So those are the specimens that we have for this year's practical experiment, like we have listed them here. So I encourage you to try as much as possible to go through this video, and perhaps you have any question, don't forget to drop them in our comment section and we will do well to get back to you on them as soon as possible. Don't forget to like our content. Please share them with your friends, let them also benefit from what you are getting. And if you are yet to subscribe, please kindly click on the subscribe button, subscribe to our channel, and don't forget to click on the notification button so you get easily notified of subsequent uploads when we put them up on the channel. Thanks very much for following us and coming along with us. We wish you success in your exams. Thanks and bye for now.